Hello everyone, welcome to ITLS Academy. So in this session we will be starting with the next topic of medical biotechnology course, that is fundamental concepts in immunology. So again in this session we will be covering the first part and then we will move on to the second part in the next session. I am your mentor Dr. Shamaita Basu. So starting with the first initial concept of immunology. Now, what do you actually mean by immunology? Okay, so immunology is basically the study of immune system and the different aspects of the immune system. Okay, now what is immune system? Immune system is how your body is defending against any kind of harmful pathogen. Okay, pathogen means anything. It can be any microorganism, it can be a bacteria, it can be a virus, it can be a fungus, or it can be inanimate objects also, like pollen grains. Okay, like mosquito bites. Okay, so any kind of substance, living or non-living, which can be pathogenic or which can be harmful for your body, that is a harmful pathogen. And your body always has an innate system. We will be talking about the different branches of immunity. So your body always has a method or a mechanism to protect you against such harmful pathogens. And as we say, your body loves you the most. Okay, you are most loved by your body. So by any means, by any method, your body will try to always combat any kind of infection. Okay, and that study is what is immunology. Okay, so as you can see over here, they are stating that the study of the physiological mechanisms which enable the body to recognize materials as foreign and to neutralize, metabolize, or eliminate them without injury to the host tissue. So this is very important. So it needs to recognize it, isn't it, to to identify or to classify something as harmful or pathogenic. Your body needs to at first identify that particular material or that particular substance, that okay, it is going to cause some harm to the body, isn't it? And then in order to, not only the body will protect you against that harmful substance, but the body will also make sure that in that process there is no harm to your body, to your own body tissues. Okay, so that is the major concept. Now moving on to immune system. Now, what do you mean by the immune system? So there's a entire cascade, entire groups of cells, tissues, different types of cells are there, different types of tissues are there, antigens, antibodies are there. Okay, I will be talking about them in details. So all together makes up the immune system. Okay, so it is a remarkable, versatile defense system that has evolved. What is defense? Defense means a system which is defending uh, your body against the harmful pathogen. Okay, defending means basically protecting, nothing else. That has evolved to protect animals from invading pathogenic microorganisms and cancer. It is able to generate an enormous variety of cells and molecules capable of specifically recognizing and eliminating an apparently limitless variety of foreign invaders. Because invader means something which is invading your body, something which is attacking your body. Okay, so there can be a different class, a different category of substances which can actually be potentially harmful to our body. And the main function of the immune system is to recognize those foreign pathogens, to identify them, also to classify, to put under which category they are coming, and accordingly exert the defense mechanism. Okay, now we will just like to focus on the like brief history of immunology, because before moving on to the main concepts, the main terminologies, or the main mechanisms of immunology, it is very important that you know a little bit about the history of immunology, from where it actually started, the roots of immunology. Okay, so whenever we are talking about history or the roots of immunology, the most important person or the most prominent person in the history of immunology will be Louis Pasteur. The you know the An image of Louis Pasteur has been given in this diagram, as we can see. So what he did was he discovered the rabies vaccine. Okay, we all know that. And what is rabies? Rabies is basically a kind of viral disease, okay, which is usually transmitted from dogs or cats. So basically they harbor, or they are the carrier of the rabies virus. And if such a dog or such a cat goes and bites a human, then the human will be infected by that particular viral infection, okay. And it is a dangerous infection till now because there is no treatment, okay. But the good thing is that we have come up with a vaccine, the rabies vaccine we have. And if a person somehow gets, like bitten by a dog or a cat, then if immediately he or she takes the vaccine, then that person is safe and secure from that particular disease 100%. So it's a 100% effective vaccine, okay. 100% is actually not right to say, it's like 90 to 95% effective, okay. So this vaccine was for the first time discovered by Pasteur, and he was the first person to administer the vaccine to a human being, which was done in the year 1885. And that is exactly what is shown over here in the diagram, in the figure, okay. So now how he actually made this vaccine? By attenuating the pathogen. So the pathogen we are talking about over here is the virus, the rabies virus, okay. So what do you mean by attenuating? Attenuating is a very important term over here, because I will be talking about in details about vaccines in the upcoming sessions of this module of this topic, medical biotechnology. So there I will be talking about in more details. So here I would just give you a brief idea, since the term attenuate has come up over here. So attenuate means weakening. Okay, now what is weakening? So you have a virus, you have a pathogen. It not necessarily a virus, it can be a bacteria or a fungus also. But over here we are talking about virus. So attenuating means you are somehow weakening that virus. Okay, now how will you weaken? So it's very important that the virus is attenuated, because otherwise what will happen, if the weakening means we are actually removing the harmful part of the virus, means that part of the virus which is responsible for causing the disease, okay. Because otherwise what will happen, if the infective part, which is known as the virion, or in some cases it's also called the prion, so if that infective part is not removed, then the virus, after entering inside the body, it will cause the disease. But that is not the the main motive or the main objective of producing a vaccine, isn't it? The objective of producing or introducing a vaccine inside the body is to induce or elucidate a immune response, so that the body prepares itself beforehand, before the person actually contracts the disease, okay. So it's very important that we remove the main infective part and keep the remaining part of the virus, so that the type of immune response that is exerted by the body against that particular attenuated vaccine is very strong, okay. And also the memory cells remain very strong. I will be talking about in details what are the different types of vaccines and the concepts about memory cells and all, okay. So moving forward, the next important milestone in the history of immunology will be that of Jenner's, okay, because what he did was he actually uh, controlled the disease of smallpox, okay. And as we all know, smallpox is now totally eradicated from the world, actually, not only from India but from the world. And only chicken pox is like occurs now, and also we have a vaccine against chicken pox too. But smallpox is something that we have eradicated, and actually smallpox was much more harmful than chicken pox, okay. People actually died from smallpox. Like the concept in those days was that if a person contracts smallpox, then it's for sure that that person will die, okay. So it was very important to come up with treatment or option regarding smallpox, okay. So what Jenner did, Jenner was again a scientist like Louis Pasteur. So in the year 1798, he noted people who had cowpox and were spared in smallpox epidemic. So basically these people, this group of people, since they already had cowpox, they were spared from the epidemic of smallpox, okay. And he took such group of people who were spared from smallpox because they suffered from cowpox, and then he inoculated those people with pus from milkmaid with cowpox. Okay, and he re-inoculated the same boy with infectious pus from a patient in the active smallpox, means a patient who is actively suffering from smallpox. He took sample from that patient and then injected it, re-inoculated the same boy. No disease state followed these inoculations, and experiment was repeated several times with great success. Means basically, what is the moral of the story? The moral of the story is that if a person is suffering from cowpox, then automatically that person is becoming immune to another disease called smallpox. So this is all an interplay of the immune system. Means the immune system is fighting, or the immune system is defending our body in such a manner, and that defense is so much strong, so much bold, and so much long lasting, that it also may occur that if my body is defending me against a particular disease, then it may also be possible that I am also becoming immune at the same time spontaneously with some other type of same disease. Okay, which in this case is cowpox and smallpox. Okay. So moving forward, Jenner's provided the first clear evidence that active immunization could be used safely to prevent an infectious disease. Okay, so that is what is active immunization. So again, I would just like to give you a brief idea about active immunization and passive immunization. Again, this is a concept of vaccination actually, which I will be talking about in details. So active means you are just giving a vaccine, or you are actually giving the antigen which is responsible for producing the disease in the body, and then the remaining job is done by the immune cells or the immune system of your body, that is hallucinating the immune response in the form of antibody or in the form of some cells, whatever it is. Okay, but what is passive immunization? Passive immunizations means you are directly injecting the antibodies, not the antigens. So your body has nothing to do with it. There's no role of your body as in immune system. Your immune system has no role in passive immunization, because directly you are being injected with the antibodies against that particular disease. Say for example, you were suffering from COVID. So passive immunization means directly COVID antibodies will be injected inside your body. So that means what? Your immune system doesn't need to do anything, isn't it? There is no job of your immune system left. So those antibodies will go in your blood and they will directly go and bind with whatever COVID antigens are there in your body, and thus you will be saved from the disease. Okay, so that is the basic difference between active immunization and passive immunization. Okay, again I will be talking about in details when I will be coming to the session of vaccines. Okay. Now talking about Pasteur again, as you can see over here, almost 70 years later, Pasteur introduced pasteurization, which is also recognized and exploited as the general principle of underlying vaccination. So at about 1900, what happened? There were the role of phagocytes and cellular immunity were elucidated. Means phagocytes were also involved. That thing was like came up for the first time, and killed vaccines were introduced. What are killed vaccines? Killed vaccines mean where you are totally killing the pathogen. Okay, you are not weakening or you are not attenuating. You are just killing the pathogen totally, and then you are injecting like preparing the vaccine and injecting it inside the body of the patient, or the like probable patient, potential patient who can be a patient. Okay, because there are many different factors also which determines that whether you will suffer from a particular disease or not. Okay, and mostly those factors are the comorbidities, as we all know after COVID, isn't it? Because those persons who were comorbid, who were having some comorbid symptoms or comorbid diseases like high blood sugar, diabetes, high blood pressure, or any kind of cardiac issues, those persons were the most probable, or they were the most vulnerable to get COVID infection. Okay, so that thing is there, and also the complement system was described. Complement system is again a cascade of proteins which are involved in elucidating immune response inside our body. And in the 20th century, acquired immunity resulted from both cellular and humoral elements, and those were demonstrated. Opsonization was described, and the term antigen came into regular use. Okay. And these are some prominent Nobel Prize winners when we are talking about immunologic research. So we have Emil von Behring, who discovered the serum antitoxins. Then we have Robert Koch, very important, who discovered the cellular immunity to tuberculosis. Then we have Elie Metchnikoff for the role of phagocytosis. We have Paul Ehrlich for antitoxins in immunity. We have Charles Richet for anaphylaxis. We have Jules Border for complement-mediated bacteriolysis. And we have Karl Landsteiner, as we all know, for the discovery of human blood groups. Now moving forward to the main subject or the main topic, just after a brush up of the history of immunology. So that is the different components of the immune system. So very important. So firstly we have innate and then we have adaptive. So innate means something which is non-specific. Okay, or you can also say innate is something which is present since birth, and adaptive or acquired is something which we acquire after uh, through our lifetime after suffering from any kind of particular disease. So it's basically a type of active immunization also, you can say. So under the innate immune system, what we have the cellular components and we also have the humoral components. Humoral components means basically the antibodies, and cellular components means the different types of cells that are involved in the human immune system. And this adaptive immune system can again be cell mediated and it can also be humoral mediated, which is the antibody. Antibodies are abbreviated as AB. So again, see, this is a more elaborate illustration of the immune system. So let's focus on here. So host defenses can be divided into innate non-specific and acquired specific. Okay. So the innate under the innate non-specific branch, we can divide it into the first line of defense and the second line of defense. Okay. So under the first line of defense, what are there? Just focus. We have physical barriers, we have chemical barriers, and we have genetic barriers. So the first line of defense is a surface protection composed of anatomical and physiological barriers that keep microbes from penetrating sterile body compartments. Okay. And what about the second line of defense? The second line of defense will have the inflammatory response, the interferons, the phagocytosis, and the complement system. So the second line of defense is a cellular and chemical system that comes immediately into play if infectious agents make it past the surface defenses. Means once it surpasses, once it can overcome the first line of defense, then the second line of defense of the body will come into play. Okay, if it's khatam in the first line of defense only, then no story of second line of defense. Okay, can you get it? Then there is no role of second line of defense. But only if the infectious pathogen, only if the pathogen is so much harmful that it can surpass the first line of defense, then only the role or the importance of the second line of defense will come into play. Okay. Examples will include phagocytosis that engulf foreign matter and destroy it, and inflammation which holds the infections in check. And talking about the acquired or the third line of defense, that is again divided into two types: naturally acquired and artificially acquired. So naturally acquired can be either active infection or passive maternal bodies. Okay, sorry, passive maternal antibodies. Okay, and the only antibody which can actually travel through the placenta from the mother's body to the fetus is IgG antibody. Okay, that is immunoglobulin G. So that is the only antibody that can travel through the placenta from the mother's body to the fetal body, and also it is present in the milk. So when a newborn mother actually breastfeeds her child, her boy or girl, that time from that milk a lot amounts of antibodies coming. Okay, so that is also a very good source of immunity for the newborn baby. Okay. uh, so that is it. And talking about the artificially acquired type, we have again active vaccinization and we have passive immune serum. Exactly what I already talked about. Passive immune serum means the antibodies, where we are directly injecting the antibodies into the body of the uh, patient. Okay, and what is the difference between active vaccination and active infection? Vaccination, do we know what is active vaccination? I have already talked about. What is active infection? Active infection says if a person has already suffered from a particular disease, that means his or her body has already produced the specific antibodies required to fight against that disease. So if second time that same person again contracts the same disease for the second time, in that case, that person will not, develop the vital symptoms of the disease. That person will not in that extent suffer from that disease again for the second time. Why? Because the antibodies are already there. The antibodies have already formed inside that person's body when he previously suffered from the same disease. So the second time that person will not suffer again because the antibodies are there and they will just come and fight against the antigen. Understood? So that is active infection. So what they are saying about the third line of defense, so they have the antibodies, the T cells, the helper T cells, the cytotoxic T cells, and also the cytokines. So the third line of defense will include specific host defenses that must be developed uniquely for each microbe through the action of specialized white blood cells, that is WBCs, and this form of immunity is usually long term and has memory. Okay. Now talking about the innate immunity, so there are some particular properties of the innate immunity or the natural immunity. What are they? It is non-specific, it is the first line of defense, and it is a result of repeated exposure. Okay, and what are the components of the innate immunity? We have biochemical components, we have physical components, and we also have some cells. So let's focus. So among the biochemical components, we have enzymes, we have different secretions, and also pH plays a very important role for immune defense. And among the physical components, we have skin is it's also known as the largest organ of our body, so it is the largest physical barrier that we have for any kind of infection. Okay, then we have cilia, and among the cells we have phagocytes and the NK cell, that is the natural killer cells. And example of some of the this innate immunity part will be burn response. Okay, because in burn what happens, the skin is totally gone, so the outermost physical barrier of the body against any kind of infection is gone, so that is a very harmful situation for that person. Okay, and as a result any burn patient is very much highly prone to any kind of infection. Okay, and that is also one of the reasons why most of the burn patients actually could not survive. They could not make it because the skin is exposed, the skin is gone, so the entire body is totally exposed to any kind of infection. Okay, so that's the one of the main reasons, or maybe you can say the only reason why most of the burn patients do not survive. Okay, like burn patients as in those patients who are like almost 70 to 80% of the body is has undergone third-degree burns. Okay, so in those cases, patients usually die, and the only reason is infection. So this is actually a diagram which explains how the NK cells are fighting against our, like fighting for our body against foreign pathogens. Okay, so this is how our NK cells looks like. So this is a healthy cell, and there is something called MHC. The full form is major histocompatibility complex. So MHC is something which basically acts as a recognizer or a receptor of a particular cell. So this NK cell will just go and bind with that MHC1, and it will be able to recognize whether it's a healthy cell or an infected cell. So similarly, uh, if an infected cell is there, MHC will not be represented, okay, because that cell is infected. So MHC will not be there, no longer be there. So the NK cell will just recognize whether a cell is infected or not, depending on whether the MHC1 receptor is there or not. So as soon as it finds a cell who is not expressing MHC1, that means it considers that cell to be infected, and it will just release some granules or some chemicals which will be, what to say, which will be elucidating the process of phagocytosis, and then it will just go and kill and engulf that particular cell. So this is actually a diagram of the human body describing the physical barriers or the components of the innate immune system. Okay, so we have lysozyme in tears, okay, which just kills the bacteria, and also phagocytes. Then we have skin, which is the largest physical barrier, as I already talked about. Then we have pH change, okay, which is mainly functioning in the stomach. We all know that the stomach is a highly acidic pH, okay, and it is very much beneficial to protect our gut, our gastrointestinal system from any kind of harmful bacterial infection. And also talking about the vagina, in case of females, the pH of the vagina is also very acidic, which is again helping to kill many harmful bacteria that can enter through the vagina inside our body. And in the saliva also we have many antimicrobial factors like lysozyme, peroxidase, lactoferrin. These are all either chemicals or enzymes or hormones. Okay. Then normal flora and fauna, always it's there inside our body. We know that the beneficial bacteria, and they are very much helpful in protecting us against the harmful bacteria. Okay. Uh, and we also have the flushing of the urinary tract, where the body is actually flushing out not only urine but also many harmful bacteria from the body. Now talking about phagocytosis, so firstly there will be adherence, then there will be engulfment, and then there will be digestion of the pathogen, which will just go and fuse with the phagosome, and then the pathogen will be engulfed. I don't think these things need to be explained in details, because this is something we all know, and I have also given a nice diagram. So these two diagrams are basically the same. Both of them are describing the process of phagocytosis only. Okay, so I will just explain this diagram because this is also the same diagram. Okay, so at first the bacteria will become attached to the membrane evaginations, which are called the pseudopodia, which is also known as the false foot, we all know. So the cytoplasm will just make those appendages which will come out, and then it will form the pseudopodia, and then the bacterium is ingested, forming the phagosome. So this is the phagosome. Then the phagosome will fuse with the lysosome. So this is the lysosome with which the phagosome will fuse, and then the lysosomal enzyme will digest the captured material. So see, it is just broken down, because digestion means breaking down larger substances into smaller substances. That is the basic concept of digestion. Okay, so this is how it is digested, and then those digested products are just released from the cell. So it is as simple as that. Okay. Now talking about the external innate defense systems, we have the structural barriers, among which the skin is the largest, as we all know, and the outer layer of the skin is more important over here, which is the epidermis. Okay, and then we also have the inner layer of the skin, which is the dermis. There we have the blood vessels, hair follicles, sweat glands. All these are actually part of the innate immune system. We have the sebaceous glands. The sebaceous glands are basically producing a oily secretion, which is called sebum. And we also have the cilia and the cuff reflex, which helps to expel microbes containing mucus. And we also have sneeze. Sneezing is also a mechanism of defending our own body against harmful pathogens. Now talking about the external innate defense systems, what we have mucus. So mucus is actually a protective secretion which is secreted by the conjunctiva of our eyes. The alimentary system means the gastrointestinal tract, the respiratory tract, and also the urogenital tract. So the mucus will consist of saliva, tears, and mucus secretions, which will wash away the invaders and contain antibacterial or antiviral substances. Uh, and there is acidity. Sweat, sebaceous glands, sebaceous secretions, means sebum. These are all acidic in nature. They are all having acidic pH of around 5.6. And the pH of the vagina is around 5, and the pH of the stomach is around 1 to 2. I think 1 is a bit very less. The ideal pH of the stomach is actually 2 to 2.5. Okay, and these any kind of acidic pH is actually not friendly to the microorganisms, particularly to the bacteria. Okay, and there are also some enzymes which are present in the skin and stomach, and also there is tear, and we also have the normal flora, means the beneficial bacteria that we have. So they compete the pathogens for attachment sites on the epithelial cell surface, and also for necessary nutrients. Okay, so these are actually some of the broad overview of the innate defense system. So talking about the internal innate defense system, so the main function is to prevent expansion of penetration. So they recognize the carbohydrates that not normally present on cells, such as mannose, and they may cause non-specific activation of white blood cells, which will lead to phagocytosis. So phagocytosis is done besides the NK cells, means the natural killer cells. Some other cells are also there which perform phagocytosis, like we have the neutrophils, we have the eosinophils, the basophils, the macrophages, the mast cells, and the dendritic cells. Okay, and also there is clotting mechanism which can entrap organisms in the fibrin clots, and we also have the complement system which can lyse cells and also enhance phagocytosis. Okay, so you can see over here I've given a diagram. So the upper part is actually uninfected normal cells. This is basically not a diagram, it is a comic. Okay, it's a fun way of explaining what is actually the thing, the story over here. So this is a uninfected or a normal cell, so there is no stimulation of the B cell. Okay, the B cell is remaining untransformed, but in this case this cell is microbe infected. Okay, or it can be a cancer cell also. So the natural killer cell will just send the signal to the B cell that now you need to transform and start producing antibodies because the body is under threat. Okay, so that is the main concept of the innate immune system. So we are left with the adaptive immune system or the acquired immune system, which I will be talking about in the next session. Okay, so I hope you like the video. Please share and subscribe and like ITLS Academy. Thank you.